This is a short presentation highlighting Biosynth's expertise and capability in custom antibody generation. We work to understand our customers' needs for their application to provide the best possible outcome. Here we will talk through two examples of antibody projects completed by Biosynth. The first case we will look at is the use of custom antibodies in the recognition of arginine methylation, a common post-translational modification that mostly occurs on glycine and arginine-rich motifs. Arginine methylation is carried out by a group of protein-arginine transferases to produce three types of arginine methylation: monomethylarginine, MMA, symmetric dimethylarginine, SDMA, and asymmetric dimethylarginine, ADMA. The development of pan-methylarginine specific antibodies is useful in identifying proteins with methylated arginine residues and providing information on signaling pathways involved in methyltransferase regulation. Producing pan-monomethylarginine, symmetric dimethylarginine, and asymmetric dimethylarginine antibodies has remained a challenge. Many polyclonal antibodies generated for protein arginine methyltransferase substrate recognition are produced using different methylated states of GAR motifs as antigens. One problem is that these antibodies cannot identify proteins that have a methylation site outside of the GAR motifs. Biosynth developed a new set of antibodies that could recognize MMA, ADMA, and SDMA substrates using a novel antigen design. How did Biosynth tackle the complexity of this antigen design? Post-translational modification-specific antibodies are typically made by immunizing with a peptide containing the modified site and surrounding sequence from a single protein of interest. This strategy could not be used in this case because the client needed the antibody to react to the modification not in one specific protein, but when found in any protein. Others had tried to generate specific antibodies by immunizing with modified amino acids directly coupled to protein carriers. This failed to generate successful reagents, probably due to the relative size of the modification compared to the carrier protein, meaning the antibodies recognized the carrier protein and not the modified amino acid. Biosynth designed a peptide construct consisting of multiple units of modified amino acids separated by spacers to increase the visibility of the modification to the immune system. The same format has been utilized in multiple instances of similarly challenging research. Biosynth's expertise in antigen design enabled the development of a panel of antibodies that could recognize more substrates than commercially available methylarginine antibodies. The overall concept went on to be used for broader applications in its future. You can read the full story in Wang's publication titled Pan-methylarginine Antibody Generation Using PEG-linked GAR Motifs as Antigens. The next case we will look at is the use of custom antibodies as reagents for the detection of a key COVID-19 biomarker. During the pandemic, there was a sudden and urgent need to develop reagents that could accurately measure levels of SARS-CoV-2 present in a patient sample. It was recognized that to reduce the spread of COVID-19, a robust diagnostic method was needed. In response, scientists across industry, academia, and public health services came together to produce a targeted protein assay utilizing enrichments and liquid chromatography, tandem mass spectrometry to capture and detect low levels of tryptic peptides derived from SARS-CoV-2 virus. Collaborating with a leading proteomics CRO, Biosynth used our Tripbodies workflow to create antibodies to tryptic fragment peptides from a prominent COVID protein. The antibodies were then used to immunoprecipitate these fragments from patient samples and measure the relative amounts present by mass spectrometry analysis. Not only were the reagents able to perform as needed, but they also went on to be useful diagnostic tools once it was established that the level of virus present had a strong correlation to the severity of a patient's symptoms. The story can be found in full in the Journal of Clinical Chemistry and Laboratory Medicine. Biosynth's tailored antibodies has supported the development of an essential protein assay developed during the COVID-19 pandemic to detect SARS-CoV-2 and an antibody panel with broader substrate recognition than commercially available methylarginine antibodies. Thank you from the Biosynth team for your attention. If you have any questions, please talk to our antibody expert, Siobhan Crittenden, and contact our sales team via the website.